Hello everyone. So we have already seen in the previous video the structure of gene, chromosome and how the genetic material works. Okay. So in this session we are going to see the chromosomal anomalies. I hope you remember the structure of the chromosome. So you know already all this we have seen, what is human cell, what is cell nucleus, what is chromosome and how the chromosome structure is. There is short arm, there is long arm, and the short arm is P or peptide for, or long arm is Q or it is G also. And as per the structure, chromosome can be metacentric where the centromere is located centrally. It can be submetacentric, it can be acrocentric, or it can be telocentric. Now here the homologous and non-homologous chromosome. Okay, so homologous chromosome, here we can see one is inherited. So in the pair of homologous chromosome, one is inherited from male parent and another one is from female parent. I told you in last class homologous and non-homologous. Okay, so now here this is the normal karyotype. You can see here in the last, this is XY chromosome. You can see, you can divide them. The classification we have seen in the previous class. Now chromosomal anomalies. So chromosomal anomalies, also known as chromosomal aberration. Any deviation either in the number or the structure in the chromosome. It can occur in autosome, it can occur in sex chromosome, or it can occur in both. You already know what is haploid, that is 23 number of chromosomes present in a gamete. Okay, and diploid, that is normal chromosome, the set of the chromosome where we can say 23 pair of chromosome or total 46 chromosome. So that is on this basis how we are going to see, we can classify the anomalies related to the chromosome. What can be the chromosomal anomalies? It can be maternal age above 35 years. It can be because of non-disjunction during meiosis. It can be radiation. It can be autoimmune disorders. Now classification. So chromosomal anomalies can be numerical anomalies or structural anomalies. Okay, what is numerical? Numerical means number. Okay, already you know. So anomalies where numbers are affected. So here you see a chromosome complement with any chromosome number other than 46 is known as heteroploid. Okay, when we say euploid, that means exact multiple of n. Okay, now first thing you already saw that what is the normal number of chromosome which is present in the gamete and in the zygote after fertilization. So we know it is either you can say 46 chromosome or 23 pair of chromosome. Okay, and other than that is known as heteroploid. It can be euploidy or aneuploidy. Euploidy means multiple of n. Now multiple of n is 46 also, but 46 is not heteroploid. I told other than 46. So then what it makes? So 69 is euploidy. Okay, because that is multiple of n. Okay, till 2n we are saying it is a normal, but if it is 3n, 69, that is euploid. Aneuploidy means here any number not exactly the multiple of n. That means here there is no 2n, but there can be additionally one chromosome or less than that one chromosome. That is known as aneuploidy. So there can be 45 chromosome, there can be 47 chromosome. Okay. Now when you say euploidy, that means I have already told you the multiple of n. Okay, but we are not saying 2n. We are saying it can be, if it is 3n, that is triploidy, or we say 4n, that is tetraploidy. So triploidy fetus are born with an extra set of chromosome. That means either it can be 69 XXX or 69XXY. What can be the cause? It can be fertilization by two sperms, or there can be diploid egg or sperm due to failure of one of the meiotic division, that is because of triploidy. Tetraploidy means, as I told you already, it is 4n. Okay, so fetus born with the two extra set of chromosome, it can be 92XXXX or 92XXYY. Okay, now aneuploidy. Aneuploidy means most common and clinically significant, always associated with physical and mental maldevelopment, or it can include both also. Why there is aneuploidy? So remember always this word, non-disjunction during meiosis, and there is unequal distribution of chromosome in the daughter cells. So that means one daughter cell will get one lesser chromosome and one will get one additional chromosome. Okay, it can happen that way. Okay, so now here you see the non-disjunction. Can you see here? So one daughter cell is getting additional chromosome, but other one is not at all getting. Here also you can see. Okay, here also you can see. So fusion with the normal gamete will happen. So here there is an additional chromosome and here there is one less chromosome. The same thing you can see here. This is non-disjunction during the meiosis. Okay, now in aneuploidy. Again, aneuploidy means there is either additional one chromosome or less than, means lesser one chromosome. So you can see monosomy, here absence of single chromosome. When monosomy happens in autosomes, it is lethal, but in case of sex chromosome, example is Turner syndrome, where there are 45 chromosomes because one X chromosome is missing, and that is why it is 45XO or X0. Trisomy, here there is an additional extra chromosome, additional chromosome or extra chromosome. Here the example of trisomy, if you say chromosome 21, there is an triploidy, sorry, not the triploidy. If I am using the word triploidy, it means 3 set of chromosome. So if additional chromosome is there, it is trisomy. So if it is trisomy 21, it is Down syndrome. Trisomy 18, it is Edwards syndrome. Trisomy 13, it is Patau syndrome. When there is trisomy in the sex chromosome, it is Klinefelter syndrome, that is 47X, X and Y. Here you can see the monosomy. Can you see? Because in your exam you will be getting these images. So can you see here? There is one X chromosome is missing. So you can say it is Turner syndrome, 45XO. So monosomy, sex chromosome, okay, where one X chromosome is missing, it is Turner syndrome. What can be the feature of Turner syndrome? You can start from above. So first thing, there will be short stature, there will be low hairline, there will be characteristic facial feature will be there, constriction of aorta will be there, poor breast development will be there, there will be elbow deformity, ovaries will be rudimentary, okay, so underdeveloped gonadal structure will be there, no maturation will be there, nevi will be there on the body, brown, nevi means brown spots, small fingernail will be there, shortened metacarpal will be there, and this one widely spaced nipple will be there, and shield shaped thorax will be there, okay. That is Turner syndrome. Now, as I told you, Edwards syndrome, it is trisomy 18. You can see here. So you can see, remember when you will be getting these charts, the chromosome number will be written there. Like this is chromosome 1, 2, 3, then 4, 5, then 6, 7 to 12, then 13 to 18. Then here X and Y chromosome. So you can see here, triploidy is there at the 18th chromosome. This is Edwards syndrome. What are the features of Edwards syndrome? You can see in the name itself, E for elongated skull, D for digits overlapping, okay, there can be clenched feet, W instead of W you can say V, V means VSD, all of you know what is VSD, A for apnea, and that becomes the cause of the death, R for rocker bottom feet, and D for dysplastic ears, okay, and there is intellectual disability. So you can see here, that is Edwards syndrome. In MCQ they will ask you So it is trisomy chromosomal 18. Then there is Patau syndrome. Now you can see here it is trisomy chromosome 13. Okay. So what are the features of the Patau syndrome? There will be major heart defect, brain or spinal cord anomalies. There will be microophthalmia. Can you see the small eyes? There will be microcephaly, small head will be there. There will be polydactyly. Can you see here? Additional finger is there. Cleft lip can be there with or without cleft palate. Okay. There can be hypotonia, episodes of temporary apnea and failure to survive. Will not survive actually. Okay. Then Down syndrome. So Down syndrome is trisomy 21. Most commonly are short note related to genetics. And you will see many of the Down syndrome cases. Okay. It can be asked as short note, as I have mentioned. Down syndrome is also known as Mongolism. You can see here there is apical fold, there is brush field spots in the eye, there will be flattened nasal bridge. In heart there will be VSD, there will be PDA, there will be ASD, there will be respiratory infection, there will be increased risk of leukemia, there will be duodenal atresia, and because of that on X-ray you will see double bubble sign. And gap between the first and second toes will be more, so sandal toes deformity or gap. Okay, so these are the feature of Down syndrome or trisomy 21 or Mongolism. Okay, then there is Klinefelter syndrome. You can see there is an additional X chromosome, so it is known as 47XXY. Okay, so we can say it is a male with an additional X chromosome. Now this becomes an example of a male having one bar body, because in my previous class I have told you bar body means number of X chromosome minus 1. So normal female there will be one bar body. Okay, but Turner syndrome it is X0, so there will be 0 bar body, and in Klinefelter there is one additional X chromosome. So Klinefelter syndrome, male with a bar body. Okay, so what are the features? Slightly feminized physique, mildly impaired IQ, tendency to lose chest hairs, female type of pubic hair pattern, there will be small testis, osteoporosis will be there, breast development happens in 30% of cases, poor beard growth will be there, and frontal baldness will be absent. Okay, now what are the structural aberration? Okay, we have seen the numerical. Now what are the structural aberration? So here the chromosome structure having the breaks followed by an abnormal combination. So at the structural level break is happening, because of that abnormal combination is happening. What can be the cause? It can be because of ionizing radiation, chemical agent or viruses. Now what can be the structural aberration? Structural aberration can be balanced or unbalanced. So balanced means there is the normal set of chromosome, and in unbalanced there is additional or missing material. Structural aberration can be stable also, unstable also. So rearranged chromosome has a functional centromere and 2 telomeres. Now structural aberration you can say balanced or unbalanced. So balanced can be example can be inversion, translocation or insertion. Unbalanced can be dicentric, ring chromosome, deletion, duplication or isochromosome. Here you can see stable in case of deletion, inversion, translocation, isochromosome, and unstable it can be because of dicentric or ring chromosome. Now deletion. Now here you see the loss of part of a chromosome or sequence of DNA during DNA replication. So any number of nucleotide can be deleted from a single base to an entire piece of chromosome. As you already know, these are the nucleotide base you would have read in the biochemistry. So any one is removed, so the whole sequence will change. So you can see here terminal deletion, it can be at one end. Okay, so single break happened, terminal part is lost. Okay, so example for terminal deletion is cry-do cat syndrome, where the deletion happens in the short arm of 5th chromosome. Okay, I hope you remember what is P and what is Q. Then there is interstitial deletion. Now here deletion is happening not at the end, but it is happening in the within the chromosome. Okay, so two breaks are there, like one break happened here, one break happened here, so this part getting lost. Okay, so intervening portion is getting lost. Two examples are there, one is Prader-Willi syndrome, another one is Angelman syndrome, and that is happening at chromosome number 15. Okay, then there is duplication. Duplication means a part of chromosome gets duplicated. You can see here these are the different nucleotides. So if you see this B and C, this part is getting duplicated, so you can see the additional genetic material. Duplication of 12th chromosome, short arm of 12th chromosome. Example is Pallister-Killian syndrome. Now isochromosome. So chromosome with the arms mirror images of each other. Okay, so abnormal split along the centromere, there is separation of the arms. Okay, so you can see here what has happened. You can see here the two arms were there, and now here what you are seeing, the split will happen in such a way that the both arms are mirror images of each other. So long arm of X, that is Turner syndrome. Okay, so this is example of isochromosome. Then there is a ring chromosome. You can see here at either end there is break, and these are fusing with each other. So a chromosome whose arms have fused together to form a ring-like structure. So denoted by symbol R in the human genetics. Two breaks are happening at the terminal end. So rarely only one end is lost, but there is no loss of telomere. Okay, so that is example of ring chromosome. Now dicentric chromosome. Dicentric chromosome means an abnormal chromosome with two centromere. You can see here one centromere, here is another centromere. Two chromosomal segment, each with centromere are fused and end to end fused with the loss of their eccentric fragment. This is also rare. Again, dicentric chromosome. Balanced rearrangement. Okay, so balanced rearrangement can be translocation, inversion or insertion. May or may not produce abnormal phenotype. Clinical importance. A carrier of balanced rearrangement can give rise to offspring with unbalanced aberration, because suppose in one parent there is balanced sorry, in one parent there is balanced rearrangement, in another parents also there is balanced rearrangement, and both are adding up and resulting in unbalanced aberration. Now what is translocation? So here you can see exchange of chromosome segment between two non-homologous chromosomes. Balanced, it can be balanced or unbalanced and denoted by T. Okay, non-homologous chromosome. You remember what is homologous and non-homologous? So both are non-homologous chromosome. In between them the genetic material is getting exchanged. That is known as translocation. Now you can see here these are two acrocentric chromosome. Okay, and there is this Robertsonian translocation. What is Robertsonian translocation? It happens between the two acrocentric chromosome. There is central fusion happens. Okay, so fragment formed by both fusion of both, and here short arm is lost. Chromosome number is 45. You can see here this is one acrocentric, this is one acrocentric, and here this short arm is lost and long arm of both are joined together to each other, and that is known as Robertsonian translocation. Okay, we are talking about translocation. Okay, so carrier of Robertsonian translocation involving chromosome 21 can give rise to Down syndrome child. Then there is reciprocal translocation. Here breakage of non-homologous chromosome will happen and exchange of material distal to break. Okay, so no loss, balanced translocation. You can see here this is before translocation. Now from here break has happened, from here break has happened. The red color came into the green color, the green color came into the red color. So that is how this is reciprocal translocation. Another translocation was Robertsonian translocation. Inversion. So here a segment of chromosome is reversed end to end and it is denoted by INV. Okay, So you can see here is the centromere. So the sequence is like A, B, C, D. Okay. So this B, C, D is getting inverted. So now it is D, C and B. Okay. So this inversion can be of two types. Either it can be paracentric or it can be pericentric. Paracentric means it is happening on the same side of the centromere only. Okay, right. Suppose this is BCD here, the same inversion happened, but it has not gone to the other arm. Okay, but pericentric, you can see here, suppose it is involving the centromere, the sequence was D, E and F. Now what happened? It involved centromere, so now it is F, E and D. Clear? This is inversion. It can be paracentric or pericentric. So in case of paracentric, there are two breaks, either P or Q arm is involved. No change in the arm ratio because it is involving one side of the centromere. So there is no abnormal phenotype. This is paracentric inversion. But here it is pericentric. Here also in each arm one break is happening. So here both P and Q arm is involved. It can result in abnormal phenotype. Most commonly inversion, pericentric type of inversion happens in chromosome 9. Now insertion. So here you see non-reciprocal type of translocation. A segment is removed from one chromosome, inserted into different one, or either in usual orientation or inverted. Here three breaks are required. So you can see here there is chromosome 4, there is chromosome 20. So here what happened? One break happened here, one break happened here. This part is removed and it is going and joining here. Okay, because here break happened, so in between it got inserted. Okay, so this is known as insertion. Example is from chromosome 4 to chromosome 20. Okay, then what is mosaicism? Mosaicism means two or more than two different chromosome complement present in one individual. Means two or more than two different chromosome setup are present. Okay, so for example in Turner syndrome, in single individual there can be 46XX and 45XO also. In Klinefelter there can be 46XX and 47XXY. Okay, and reason for that is non-destruction in early post-zygotic mitotic division. So this is mosaicism and the individual is known as mosaic. So summary of the chromosomal aberration: it can be numerical aberration, it can be structural aberration. Okay, in numerical aberration you know what is euploidy, what is aneuploidy. Okay, euploidy means the multiple set of chromosome, aneuploidy means additional or lesser number of chromosome. Okay, euploidy example that can be triploidy, heteraploidy, aneuploidy I told you. There can be monosomy, there can be trisomy. Structural aberration can be balanced when the overall genetic material is not disturbed. It can be inversion, translocation, insertion. Okay, unbalanced can be deletion, duplication, ring chromosome, isochromosome, dicentric chromosome, where either there is addition, increased amount of genetic material, or genetic material is reduced. And here is structural aberration. It can be stable also, unbalanced, and it can be unstable also, not unbalanced, unstable. So stable can be inversion, translocation, deletion, isochromosome, and unstable can be dicentric chromosome or ring chromosome. Okay, so that is the topic. Thank you.